Physical science performance task.
Hello everyone, I'm Jamaila and I'm with Ron Jeffrey Roybal, and now we will talk about everything you need to know about compound microscope.
A compound microscope is a microscope that uses multiple lenses to enlarge the image of a sample. Compound microscopes usually include exchangeable objective lenses with different magnifications and mounted on a turret. To adjust the magnification, a compound microscope uses two or more lenses to produce a magnified image of an object, known as a specimen placed on a slide at the base.
The characteristics of a compound microscope. Two or more convex lenses, typical magnification range between 40 times and 1000 times. One object is used at a time. Two-dimensional images. Available in monocular, binocular, trinocular, and multi-head configurations.
A Dutch father and son team named Hans and Zacharias Janssen invented the first so-called compound microscope in the late 16th century, when they discovered that if they put a lens at the top and bottom of a tube and look through it, objects that on the other end become magnified.
These are the parts of compound microscope. First is the head or the body. The head or the body houses the optical parts in the upper part of the microscope. Second is the base. The base of the microscope supports the microscope and houses the illuminator. Third is the arm. The arm connects to the base and supports the microscope head. It is also used to carry the microscope. Fourth is the eyepiece or ocular. Eyepiece or ocular is what you look through at the top of the microscope. Typically, standard eyepieces have a magnifying power of 10 times. Optional eyepieces of verifying powers are available, typically from 5 times to 30 times. Fifth is the eyepiece tube. Eyepiece tube holds the eyepieces in a place above the object lens. Sixth is the objective lenses. Objective lenses are the primary optical lenses on a microscope. They range from 4 times to 100 times and typically include 3, 4, or 5 of lens on a most microscope. Seventh is the nosepiece. Nosepiece houses the objectives. The objectives are exposed and are mounted on a rotating turret so that different objectives can be conveniently selected. Eighth is the coarse and fine focus. Coarse and fine focus knobs are used to focus the microscope.
Stage is where the specimen to be viewed is placed. Stage clips are used when there is no mechanical stage. Aperture is the hole in the stage through which the base transmitted light reaches the stage. Illuminator is the light source for a microscope, typically located in the base of the microscope. Condenser is used to collect and focus the light from the illuminator onto the specimen. Iris diaphragm controls the amount of lighting reaching the specimen. Condenser focus knob moves the condenser up or down to control the lighting focus on the specimen.
Compound microscope work by turning the revolving turret so that the lowest power objective lens is clicked into position. Place the microscope slide on the stage and fasten it with the stage clips. Look at the object lens and the stage from the side and turn the focus knob so that the stage moves upward.
A compound microscope is a high magnifying microscope that is used to examine the smaller samples that are not visible to the naked eye, such as diatoms, bacteria, or viruses. In this microscope, the sample is kept in a glass slide and then is kept on a stage on the microscope to examine through the lenses. Compound microscopes allow scientists to see microorganisms and cells. Without these microscopes, we would not know about the existence of cells, and therefore, we would not able to study DNA or make medical advances based on our knowledge of how different diseases or the condition of attack cells.